Hi everyone, I'm S.Sahabudeen, the head of the Department of Biotechnology. Our department has started in the year 2003 and it has grown out to be one of the leading departments in our university. In this department, we have one distinctive lab called the Developmental Biology Lab, established by the funding of the DST ECR funding. And we also have an industrial sponsored lab called the EBPSR MIST lab. EBP is an international consultancy industry located in Tokyo, Japan. So these labs, both the labs, concentrate on environmental aspects where we face a lot of challenges by pollution in air and water and in our food. So we work for the problems and we address solutions that are needed by the society. In addition, we also take up research problems that are requirement and are the pressing needs of industries and collaborate with the industries to run joint projects that can give products that are important for the industries and to the society. You all are welcome to look at our research and our patents in this IRIS 2025. Welcome to IRIS 2025.
Hi, I'm Sharine. I'm pursuing my PhD in the Developmental Biology Lab. I'll be providing a brief overview about the various research scope in our lab. We primarily focus on toxicological studies using the Drosophila melanogaster model. We intend to identify environmental toxins and test it on this model. Leveraging both wild-type flies and transgenic models, we intend to find out the genetic and molecular patterns of these toxic compounds. So since Drosophila is a convenient and an easy model to handle, we'll be able to extrapolate these data to human health concerns because of its genetic similarities with humans. In this regard, we have acquired 2 patents. We've identified potential therapeutic compounds that can mitigate acrylamide, a potent environmental neurotoxin. Further broadening our research area, we are also interested in identifying microplastic toxicity and also for screening of antimicrobial resistant organisms and detection of viruses in the environment. So in this regard, we have established a memorandum of understanding with the industries who help us in this investigation process. Furthermore, we are also committed to maintain a sustainable environment. So our research also focuses on bioremediation of plastics and also produce sustainable alternatives to the conventional plastics. So to summarize, our aim is to bridge the fundamentals of toxicological insights with practical solutions to foster a better and healthier sustainable environment. Thank you.
Hi, my name is Swayam and the project which we are working on is isolation and identification of antimicrobial resistance organisms in wastewater samples. So, what AMR is? AMR is basically any ability of microorganisms to withstand the effect of antimicrobial drugs or antibiotic anything. So, what our project mainly focuses on is isolation, identification and characterization of these antimicrobial resistance organisms from wastewater using various molecular and biochemical assays. So, the insights coming from our project will mainly help us to understand the spread and the implications of public health of these microorganisms.
Okay, so hi, my name is Ria.
And I'm Aditya.
So our group is basically working on tackling the problem of plastic pollution. So plastic, as we know, is becoming a cause of great concern as it is causing a lot of harmful impacts on the marine as well as the terrestrial environments. So we are this is because of the excessive production of plastics as well as its non-biodegradable nature, which causes it to persist in the environment. So hence we are looking to create bioplastics, which is an eco-friendly alternative to reduce the over-dependence on single-use plastics.
To produce bioplastics that are eco-friendly, we have decided seaweed as our source material. We have collected a lot of seaweeds and identified them. Through our research, we have produced and developed a method to produce bioplastics. And through continuous research and experimentation, we are optimizing our approach to produce a better alternative.
So hi, this is Tanmay, a 4th year biotechnology student working in Developmental Biology Lab. So we are assessing the toxicity of nanoparticles using the Drosophila melanogaster as our model organism. So the reason why we chose this model organism, because it has 75% genetic similarity, especially in the disease related aspects in the fly. And in laboratory conditions, we culture our flies in flask with specialized enriched medium. It's a very essential and an effective model to assess the toxicity in this model, because because of the ease of maintenance, and also we can conduct the huge large scale experiments in a short period of time. Yeah. Thank you.
I am Mitran,
I am Sushit Vivedi.
and our research work is based on wastewater treatment using plant-based compounds. Here we are using biochar, which is obtained from plants for wastewater treatment. Biochar is generally porous in nature, so that it can accumulate various pollutants like microplastics, dyes, pharmaceutical waste, etc. So now, after adding biochar in wastewater and letting it to react, we can simply separate the treated water by simple sedimentation and separation. With this treated water, we are carrying out toxicity studies using Artemia salina, which is a marine organism. So what is our outcome of our project is, our treated water should be safe so that we can directly let it to the marine environment.
So through the toxicity studies, we are trying to see that the Artemia salina are able to thrive in the treated water which we obtain from the biochar treatment. Through this, we can see that the treated water is able to mimic the marine environment, and also the aquatic organisms are not harmed by it, so that we can release the treated water into the aquatic bodies normally.